Okay, so let's talk about enzymes, which are a special type of protein. So, quick review of what a protein is. A protein is a folded polypeptide, which is made up of a monomer of the monomer amino acids. So remember our macromolecules, you had lots of amino acids. Amino acids are the monomer or the beads. Okay, they're tiny little molecules, and when you add them all together in a certain order, remember it depended on which order you put them in. There were 20 different types of amino acids. And they all bind together through something called a peptide bond. So when you bind them all together, you get a protein, and usually they're very, very, very long. I don't know, hundreds and hundreds of, maybe thousands of amino acids all bound together. When they're formed, they fold in a certain way. So this first picture that we had, this is a model of a certain type of protein, an enzyme, and all of these different little like squiggles and things, that's supposed to represent how it folds. So because of the different interactions, you don't really need to know great detail about it, but because of the different interactions of these beads, these amino acids, the protein will fold in on itself and create some sort of structure. So, it could create a little squiggly thing like this. Usually they, usually they just look like a whole huge mess of squiggles. Let me show you another picture. Models that show that. It's very hard to get, most of these pictures are models of these molecules because we're talking about atoms, like lots of atoms together, but still atoms that are very, very small. So these are the type of pictures that we have of them. So they fold together, and that creates something called a protein, which are one of our macromolecules. All right, so let's talk about very quickly chemical reactions. So you know what a chemical reaction is? It's when different compounds come together and they either form a new type of molecule or a new compound, or they get broken up into different types. So with a chemical reaction, you have reactants, which are the starting materials, and then you also have the products. So what forms after the reaction? If you take chemistry next year, you'll learn all more about this, but just for our purposes, again, you could have something like a reactant would be if you have compound A plus compound B, and you add them together, then you would get compound AB. Or maybe you could start off with something that is already stuck together, AB, and you go through a chemical reaction, maybe catches fire, or you add acid to it, or change the temperature, something like that. Could be a lot of different things. And it forms A plus B. So that's what we mean by when the bonds of atoms are either broken or formed. This first one, they would be formed. You have two separate ones, the reactants, and then the product would be when they're stuck together, or if they're broken. So they were stuck together and then they go back to being separate. So to start a chemical reaction, like everything in biology, you need energy. Energy has to be added. The type of energy, or what we call the energy that needs to be added to start a reaction, is called the activation energy. Activation energy. Amount of energy that needs to be added to start a chemical reaction. You could think of it as the chemical reaction would be, so this guy's pushing a ball up the hill, and the chemical reaction, like either forming of those two elements or breaking of those two elements, would be the downhill, would be the ball rolling down the hill. The activation energy is the energy that this guy is using to push that ball up the hill. Okay, lots of energy involved to just get it up, and then once you actually got all the energy you need, chemical reaction would be boop, it just falling down. Energy and activation energy. Enzymes are important because they speed up this chemical reaction. They make it so that you don't have to put so much energy into the system to get it going, which is very, very important because We have lots of, think of our bodies, we've got like just breathing takes a plethora of energy and chemical reactions happening. Okay, you're taking in you're taking in oxygen from the environment, from the world, the atmosphere, and your body is going through all these crazy chemical reactions, cellular respiration, which we'll learn about in the next unit, inside of your lungs, and then out pops CO2 and energy and things like that, okay? So again, enzymes are a type of protein. They allow chemical reactions to occur by increasing the rate of the reaction, and it's needed for everything, almost all, well, I said everything, but almost all processes in your body, in all living things. In plants use them. Plants need enzymes. Bacteria need enzymes. Fungi need enzymes. Humans, dogs, literally any living thing needs enzymes to do this. Hemoglobin, pretty important for living, right? Pretty important for carrying oxygen around in your blood. Otherwise, we wouldn't be able to do anything. Without enzymes, just creating a hemoglobin would take billions of years. We don't have that time, okay? So that's why enzymes are so important. Again, enzymes are a type of protein that are folded all over in a certain way. So basically, the way that they work is they just make like a landing site for whatever products need to get together or whatever products need to split up. So we call that active site of the enzyme. The substrate would be basically the reactant. So we say substrate also mean reactant. Okay, it's a landing site for these chemicals to land on and then break apart or either form new bonds. It's a location on an enzyme where the substrate binds and reacts. Again, these are all models of, but just imagine like a crazy, something like this, crazy fold type of molecule that has a little site in it that fits perfectly for whatever it is that is for whatever biological process you need. All right, so substrates, we also would call this reactants. They would fit perfectly like puzzle pieces into the specific enzyme that is needed. The enzyme is the landing site for these two, helps them get together, and then you have the product. Otherwise, these substrates, if you didn't have this enzyme here that is like kind of coaxing them to come and get together and speed up this process, if you didn't have this here, they could just be floating around in your body or bloodstream for who knows how long, and maybe they'll randomly get together, but the enzyme is kind of like, come on, let's do this, let's get going. So that's what we mean by when it lowers the activation energy. It just makes it easier for these things to get together and create your products, whether that's hemoglobin or something else. So this showed, if you had two substrates or two reactants that are trying to bond together. So we were talking about with the beginning, we had A, get rid of that. If you had A trying to get together with B, then you would create product AB bound together. Or if you already had two that were together, AB, and the enzyme needs to split them up, A plus B. Enzymes can do both of those things. So again, how do they speed up reactions? They lower that activation energy. They provide a place for both substrates to meet and react, and they provide favorable conditions for the substrates as well. So, this slide of the properties of enzymes. They're not used up in chemical reactions. They are recyclable, so they're used over and over and over and over and over again. They're the same landing site for all sorts of reactant, or for the reactants, okay? It doesn't just get done once. They are used over and over and over again. They lower the activation energy. They increase the reaction rate within your body, or the bodies of living things. And they're specific to only one substrate. So you have one specific enzyme for creating hemoglobin. You have another specific enzyme for, helping you with cellular respiration and breathing. Enzymes and substrates fit together like a lock and a key, or like puzzle pieces. Identifying enzymes, pretty easy. They all have ACE at the end of them. So catalase, sucrase, lactase, DNA polymerase, kinase. They're all examples of enzymes. So if you notice anything, they all end in ACE, A-S-E. DNA polymerase is an enzyme we'll talk about when we get to genetics. It's a very important enzyme for all of our cells because it helps DNA unwind and get copied and get, and create all of our genes and things like that. So when enzymes don't work, it can be an issue. And this happens if there are, if the pH, maybe it's too acidic or too basic. Remember we talked about that. Or if it's too hot or too cold. Okay, so enzymes are finicky, as in they only really function best in a small range of conditions. So the optimal, maybe it's pH. Maybe they only work really, really well if the pH is slightly basic, it has an 8 pH, okay? It just depends on what type of enzyme it is and what we're talking about. Like all proteins can go through something called denaturing, and this is what happens when the enzyme changes shape or loses its function. It's not just enzymes that do protein. Any protein can go through denature, and what that means is it like unfolds. One of the most common examples of this would be when you cook an egg. All right, what do? How did it look before you started heating it up? It was gooey and not white, and if you ate it, it would make you sick. So we actually need to denature this type of protein if we're gonna eat it and make it safe for us. But what are you doing? You're heating it up and you're causing a chemical change in all of these proteins, in all of these chemicals, or all of these molecules that make up an egg. When you heat it up, you are causing them to unfold in a certain way. Once something is denatured, you can't put it back, okay? So remember we said enzymes are specific to substrate, fit together like a lock and key. If the shape changes, then the substrate will no longer fit. So you could think about denaturing as like you had a puzzle piece that looked this way, and perfect little heart puzzle piece would fit into it, but if it got warped, if it got too acidic or too basic, and that, suddenly now it's stretched out and it can't fit there anymore, so it doesn't work. We already talked about how if something is too hot or too cold, or if it's too acidic or too basic, that is what can denature an enzyme, and not just an enzyme, but a protein as well, because enzymes are proteins. So that concludes all of the notes for this unit, but let's just do a brief summary of everything that's going to be on your Unit 2 test, which Unit 2 was over biochemistry. So the first thing that we talked about were our macromolecules. The majority of this unit are your macromolecules. So those were carbs, carbohydrates, lipids, proteins, and nucleic acids, DNA and RNA. Okay, we just did, so these notes were just over enzymes, which are a type of protein. Okay, most of your test is going to be over macromolecules. That's what your quiz was over. Making sure that you know the monomer of each, so making sure you know the monomer names versus the polymer names of every single one, and then also the function of them and what they look like, so their diagrams and pictures. Next, we talked about properties of matter and water. Mainly properties of water, but you do need to know the different types of bonding. So covalent bonds versus polar bonds versus non-polar bonds, why water acts the way that it does because of its hydrogen bonds and because that it's such a polar molecule. Okay. And then the last thing that we talked about were enzymes. I'm sorry, that's not the last thing we talked about. We also had pH. So properties of matter and water, pH, and then enzymes, which are a type of protein. So, you know, how it works, how it lowers the activation energy, things like that. Also need to know about the pH scale and, you know, what's an acid. Remember that was for acid versus base. What makes something acidic and what makes something basic. So this is everything that is on your Unit 2 test.